Ah, so the third experiment is performing Alcian blue stain. So what is the reason for this Alcian blue stain? So basically, students, we have only two stains that are actually okay. We have only one stain, okay, which can that has properties to stain the mucopolysaccharide membrane. Okay, usually this Cryptococcus neoform's capsule, it is really challenging to stain Cryptococcus neoform's capsule. Okay, if you want to stain that particular capsule means, then we need a specialized stain, and that specialized stain is our Alcian blue stain. So just a second, I don't know why I lost my PowerPoint. अरे यार. Hmm. Okay, students. So listen to me. Am I audible to you? Am I audible to you, students? Anuj, Ashish,
Yes, yes.
Indrajit, Kajal. Fine. Okay. Listen to me. Um, what is the purpose of staining? Can anyone say? Why we will stain the cells or tissues? What is the purpose of staining? Can you give any answer? Why we are staining?
DNA.
To visualize under the microscope. To visualize the cells under the microscope, we will stain the cells, right? Now, um, can we able to detect specific components in the cell? See, like I have a glycogen in the cell. I want only, I need to stain only the glycogen part of the cell. I don't want to stain complete cell. Rather than I want to stain only glycogen. I want only the glycogen to visualize in the cell. Is it possible like that? Maybe it is possible. How means I will create a color substance which will have attraction towards the glycogen, specific attraction to the glycogen, so that by this stain I can able to target glycogen in the cells. Now this became a glycogen specific stain. So by creating specialized stains to visualize specialized parts of the cells, will have an advantage. That advantage is, rather than seeing the cell, now we are seeing a specific part of the cell. So for example, in case of multiple myeloma, the patients will have some ground substance. Some ground substance will be deposited in the tissues. This ground substance can be visualized by using Congo red stain. So by using Congo red stain, we can able to visualize this ground substance. Presence of this ground substance in Congo red stain indicates that the patient has multiple myeloma, or there is a glycogen storage disease in the liver. There will be excessive storage of glycogen, results in liver damage. Here, I will prepare a stain which will attract towards the glycogen part of the cell. So if I'm getting color out of this particular stain means, by that I can conclude that the patient has glycogen storage disease. Such stains are called special stains. Special stains have some special purposes. They have specific targets in the cells to stain a specific component. One of the specialized stain is our acid fast stain. Acid fast stain only stains the Mycobacterium species. It won't stain any other species. The acid fast is an example of special stain. Similarly, Why can't we use a specialized stain that has the properties to attract towards the mucopolysaccharides? We know that mucopolysaccharides are really challenging to stain, but why can't we prepare a stain that has specific properties to attract towards the mucopolysaccharides? If we prepare that special stain means that can be used to identify Cryptococcus neoforms. Why? Because traditionally we will go with negative staining to identify the Cryptococcus neoforms. The problem with negative staining is even fat blob, even a small fat bubble will be falsely identified as Cryptococcus neoforms, because even fat also won't stain. Background will be stained and fat will be stained like this, and we will think it is Cryptococcus, and it will give false positive report. So rather than keeping our trust on the negative stains, better trust the special stain. Why can't we visualize the mucopolysaccharide part of the cell? To stain the mucopolysaccharide, we have this pastille. So this pastille or Alcian blue stain. So this Alcian blue stain will specifically stains the mucopolysaccharide layer of the Cryptococcus neoforms, or the capsule of the Cryptococcus neoforms can be stained with the help of this Alcian blue stain. Okay, so this is our third experiment. So similarly, we have many special stains. We know that we have multiple cells in our body, multiple tissues. We can able to target, or we can able to prepare special stains to visualize a target tissue in the body. Such stains are called special stains. Okay. For example, we have Congo red stain, Wang-Yin stain, Manson's trichome stain, reticulin stain, then oil red ore stain, periodic acid shift stain or PAS stain, Alcian blue stain. So these are multiple special stains that has properties to stain a specific part of the cell. Similarly, we have the Alcian blue stain, which has the special properties to attract towards the mucopolysaccharides. Okay. So I'm not going in depth about the structural classification of mucopolysaccharides. You already know mucopolysaccharide is a type of carbohydrate which is glucosamine glycan. Okay, I'm not going this much depth. So you just remember mucopolysaccharide need to be stained. This mucopolysaccharide is present in the capsule of the Cryptococcus neoforms. So forgot about the mucopolysaccharide structure. We are not worrying about that. We are only concerning about staining the mucopolysaccharide. That's it. Okay. So as I said, Cryptococcus neoforms will cause this brain infection. So it will literally eat your brain alive, and the patient will go to coma, and the patient may go to death. Okay, a brain dead. So one of the deadliest fungus-mediated infection, fungus-mediated systemic infection. Okay, so that is caused by our Cryptococcus neoforms, and this Cryptococcus neoforms is a capsulated fungi. That means its capsule is made up of mucopolysaccharides, and it is really challenging to stain a mucopolysaccharide membrane containing cells. So that is what traditionally we will go with Indian ink. So in the Indian ink, we can able to visualize the mucopolysaccharide or the capsule of the fungi. But to confirm that this is an actual Cryptococcus neoforms, then we have a specialized stain called Alcian blue stain. So in the Alcian blue stain, if you are getting positive result means we can confidently give report as positive for presence of Cryptococcus neoforms in the patient. That is why we are relying on Alcian blue stain to give a confirmation report to the patients. Okay, so Alcian blue stain is a specialized stain that has affinity towards staining the mucopolysaccharide part of the capsule of the Cryptococcus neoforms. So today's aim is to perform Alcian blue stain for identification of acid mucopolysaccharide containing fungus such as Cryptococcus neoforms in the given sample. So today we are going to visualize Cryptococcus neoforms that contains acid mucopolysaccharides by using Alcian blue stain. That is the aim. So the principle is very important. How we managed or how we developed this particular stain with specific properties of showing affinity towards the mucopolysaccharide. That is the real tricky part of this particular test. You need to know the mechanism of this Alcian blue stain. What is the mechanism? Just a second, students. Now I will give you a statement. Kindly listen it carefully. We can you able to stain a cell with the help of turmeric powder water? Take a water, mix some turmeric powder so it will become yellow solution. Can you use this solution to stain a cell? Is it possible? It's not possible. You cannot stain a living cell with the help of turmeric powder. That is not stain actually, it is a dye. Dye can only apply to inanimate objects, but dyes cannot be applied to living cells. To stain a living cell, you must charge the stain, you must charge the dye, you must introduce some charge to the dye. Then only the dye can able to bind with the living cell. Usually living cells will have this negative charge on their plasma membrane. And our stain should have a positive charge. Then only our stain will attract towards the negative charged cell membrane. The same phenomenon applies to our Alcian blue stain also. We need to ensure the charge of the stain remains cationic in nature. That means the stain charge, electric charge should be positive. Only in positive charge, the stain can able to bind with the mucopolysaccharides. Now, there is one important point. That is, there are two types of mucopolysaccharides. One is acid mucopolysaccharides, and second one is sulfonated mucopolysaccharides. Sulfur containing mucopolysaccharide and acid containing mucopolysaccharide. This sulfur containing mucopolysaccharide and acid mucopolysaccharide. These two can be stained at different pH. Sulfonated mucopolysaccharide can be stained at pH 1, whereas carboxylated mucopolysaccharide can be stained at pH 2.5. pH 2.5 is for staining carboxylated mucopolysaccharide. pH 1 is for staining sulfonated mucopolysaccharide. These two can be, if you want to stain only sulfonated mucopolysaccharide, then ensure the pH of the solution is around 1. Then you can target the sulfonated mucopolysaccharides. If you want to stain carboxylated mucopolysaccharide, then your pH should be around 2.5. So this is the basic idea. Okay. Then coming to the principle. So in the principle, very simple, students. This particular stain has a central copper ring. It has a central copper ring. Okay. Now this central copper ring is connected to a structure called isothiocyanate. Okay, this is called just a second. Phthalocyanine ring. So this central copper ring, this central copper atom, this copper atom was surrounded by a phthalocyanin ring. So this ring is called phthalocyanin ring. So PTHALOCYANIN, phthalocyanin ring. As the name suggests, it contains cyanide. Okay, it's a derivative from cyanide. So central copper atom was surrounded by a phthalocyanin ring. Now, in the each corner of this phthalocyanin ring, it will contain positive charged substance. That positive charge is given by this positive charge was given by isothiouranum groups. So isothiouranum groups will give the positive charge. These are isothiouranum groups which are connected to the phthalocyanin ring. How this isothiouranum attached to the phthalocyanin ring? It is attached to phthalocyanin ring by thioester bonds. With the help of thioester bond, this isothiouranum was connected to the phthalocyanin ring. Okay. Now, this isothiouranum will bind to the negative charged mucopolysaccharides. This isothiouranum will bind to the negative charged mucopolysaccharides. By what bond? By what and uh, by what energy? Okay, that is dependent upon electrostatic bond. The bond between this positive charged isothiouranum and mucopolysaccharides will be electrostatic bond. So electrostatically, this isothiouranum will bind to the negative charged mucopolysaccharides. At pH 1, it will bind to the sulfonated mucopolysaccharides, whereas at pH 2.5, it will bind to the carboxylated mucopolysaccharides. So this is the principle of Alcian blue stain. So Alcian blue stain will have central copper ring, which is surrounded by phthalocyanin ring. This phthalocyanin ring has four corners. In the four corners, it has the isothiouranum. This isothiouranum molecule binded to the phthalocyanin ring with the help of thioester bond. Then this isothiouranum bind to the mucopolysaccharides with the help of electrostatic bonds, which is a reversible bond. Okay, so that's it. This is the principle of the Alcian blue stain. Then for all special stains we need a control. So in case of here, the control will be small intestine, appendix, or colon. So control means this is a known sample. So we can use small intestine, appendix, or colon as a known sample, which can be used as a control for this test. Then materials required are coupling jars, beakers, some glasswares for preparation of these reagents. So we need to prepare two types of solutions. First solution will have pH 1 for staining sulfonated mucopolysaccharides, and second solution will have pH 2.5 to stain the carboxylated mucopolysaccharides. To prepare pH 1 Alcian blue stain, all you need is Alcian blue solution at pH 1. Then you need to add 1 gram of Alcian blue stain to 8GX. 8GX is a component which is readily available in the laboratories. So you need to take 1 gram of Alcian blue 8GX to 100 ml of 0.1 normal HCl solution. If you add 100 ml of 8GX Alcian blue, 1 gram of Alcian blue to 100 ml of 0.1 HCl, then you will prepare your pH 1 Alcian blue stain. If you want to prepare 2.5 pH Alcian blue stain, then you should use acetic acid as a acidifier to adjust the pH. So boil 100 ml of distilled water and add 3 ml of concentrated acetic acid to it, and add 1 gram of Alcian blue AgX dye. Then it takes a lot of time to dissolve the Alcian blue stain. Mix and adjust pH 2.5 by using acetic acid. So with the help of acetic acid, you can adjust the pH 2.5 and you can use it. So you can store this solution at room temperature for 6 months. You can use almost for 6 months. Okay, so that's it. So these are the procedure. So the procedure is very simple. First, you need to deparaffinize the tissue. Why we are deparaffinizing? Because we will get brain tissue samples. That is why brain biopsies. So in the brain biopsy, you will have that paraffin wax. So that paraffin wax need to be removed. Then you need to stain with Alcian blue for 30 minutes. Then wash underwater. Then counter stain with hematoxylin solution for 1 minute. Then wash underwater. Then dehydrate with alcohol, clear with xylene, mount in DPX, and observe under the microscope. In the microscope, what you will see is you will Alcian blue stain acid mucins will be in bright blue color. Presence of bright blue color indicates presence of acid mucopolysaccharides. That's it. So this is the results. So very simple test. I'm telling again, Alcian blue stain is used for staining acid mucopolysaccharides or mucopolysaccharides in the fungal cell wall. That can be done by using this Alcian blue stain. Alcian blue stain has a central copper ring with phthalocyanin ring. It is connected with at the four corners of this phthalocyanin ring. It has isothiuranium molecules. This isothiuranium molecules bind to phthalocyanin ring with thioester bonds, and this isothiuranium molecule bind to the acid mucopolysaccharides via electrostatic bond. Okay, solutions we will prepare two. One is pH 1 and other one is pH 2.5. For 2.5 we will use acetic acid. For 1 we will use 0.1 normal HCl solution. That's it. Procedure is you need to defaragnize the tissue. You will primary stain with Alcian blue for 30 minutes. Then you will run under wash tap water. Then you will counter stain with hematoxylin. Then you will wash under tap water. Then you will dehydrate, clear and mount with DPX, and you can observe under the microscope. In the microscope, you will see mucopolysaccharides in the bright blue color. That indicates positive for the test. That's it. This is all about the Alcian blue. If you have any doubts, you can ask me. Obviously, you will have doubts. The only doubt will be the principle that you can go through the notes. Okay. So principle is very simple. It has a central copper ring surrounded by a phthalocyanin ring. It is at the four corners. It has isothiuranium molecules which are bind to the phthalocyanin ring with thioester bond, and this isothiuranium molecule bind to the mucopolysaccharide electrostatically, which is a reversible bond. That's it. I will share this lecture video also. Kindly go through it for revision. So thank you very much, students. If you have any doubts, you can ask me. If not, I will end this session. Can I end this session? Your class finished. Can I end this? Any doubts in this lecture?
The practical file submit here.
You need to submit your practical files in the MS Teams. I will create right now. I will create an assignment and you submit in the MS Teams itself. Okay? Practical file. I think Anuj already finished complete record, it seems. Okay. So thank you very much, students. Submit in MS Teams. Okay? I will make it right now. Thank you very much.
Thank you, sir.